Hello everyone, this is Mr. Furman again. We're going to continue our notes today on protein synthesis. Today I actually want you seeing two videos. The first video we're going to go through the majority of translation. The second video, there isn't really any notes with it. I just wanted to give you a visual to go with translation because this is a process that may be hard to visualize. So to begin, let's do a little review from yesterday and let's talk about transcription. Remember, transcription is a process that occurs in the nucleus that creates a usable copy of the information on DNA, which is known as messenger RNA or mRNA. mRNA, remember, is written in the nitrogen bases A, G, C, and U. Uracil replaces thymine. And when you look at this, you have DNA, and the product that you get from it is mRNA. And you can see the difference that mRNA is also single-stranded. Now, the last part of transcription leads to what mRNA can do. mRNA can leave the nucleus. Remember, DNA can't. And mRNA can go to the cytoplasm through small holes called nuclear pores. DNA can't fit them, but you can see here the mRNA would be able to fit through this small nuclear pore. Okay, what happens after transcription? Well, mRNA is now in the cytoplasm. That workable copy is in the cytoplasm. Think of it from yesterday. I talked about how DNA was this master cookbook and the mRNA was a copy of a recipe. You've now gotten the recipe into the kitchen. Think of the cytoplasm as a kitchen. This is where all the protein synthesis, the creation of proteins occurs. Now, how do we get that information? If you look at an mRNA, it's written in nucleotides, and proteins are written in amino acids. So mRNAs are written in nucleotides, proteins are written in an amino acid sequence. They're two different things. Well, this is where we go through a process to translate from nucleotide to amino acid, hence the name translation, where you translate the information in mRNA to make a protein. So, when we look at this, the area that gives us the information on the mRNA is what's called a codon. And we're going to talk about these codons a little bit more. Now, codons are a group of 3 nucleotides on mRNA. And those 3 nucleotides, those codons, actually code for 1 amino acid. So if you look at this, if these were 3 nucleotides, these 3 right here would code for 1 amino acid. Same thing here. And same thing here. So like in the last example that I have, the codon GGG codes for the amino acid glycine. Kind of like if you saw something in your recipe book that called for lemon juice, you would get lemon juice now. You wouldn't get anything like a lime juice or an orange juice or whatever. So it gives specific instructions of what recipes are needed. Now, how many codons are possible? Well, you have to look at it. Remember, a codon is made up of 3 nitrogen bases. The nitrogen base could either be A, G, C, or U. Same thing with the second one, same with the third one. So we have 4 possibilities times 4 possibilities times 4 possibilities. So we have 64 possible codons. Well, 64 possible codons, but there's only 20 different amino acids. Well, what does that mean? Well, more than one codon can code for the same amino acid. For example, GGG, GGC, GGA, GGU all code for the amino acid glycine. So how do I know what amino acid goes with what codon? Well, you have to look at this chart. I'm going to give you kind of the complex version right now, and I'll give you an easier one, most likely on Wednesday or Thursday. But it's this chart right here, where it gives you a certain type of amino acid based on the type of codon you have. Now, let's figure out how to read this thing. Well, remember, codons have 3 nitrogen bases. The very first one is called the first nitrogen base. The one in the middle is the second nitrogen base. And the one on the end is called the third nitrogen base. You use that chart that I showed you to determine the amino acid. The first nitrogen base, you look at the left column. The second nitrogen base, you look at the top row. And the third nitrogen base, you look at the right column. So what do I mean by that? Let's take a look at this and do a little practice with this. So let's say we're looking at the amino acid UGA. Well, U is the first nitrogen base. We're going to go to the first codon. It's U. So we know that it's one of these amino acids right here. Well, you look at the second one, G. So it's U and G. It's got to be one of these four right here. Then you go to the third one, A, UGA. It's a stop codon. And we'll talk about what a stop codon is here in a second. So U is the first one, to recap. G is the second one, and A is the third one. So you get UGA, the stop codon. Now, a little practice with this. I'm going to give one here. You can stop and look at it and see if you're right. So let's say I wanted you to find what amino acid would be with CGA. So pause the video right now and try and figure it out. Okay, CGA. Well, the first codon is C. First nitrogen base is C. G, so it's in this one. A, CGA, would be targeting. Okay, we talked about, in the first example, I said a stop codon. Well, some codons have special instructions. They don't make an amino acid per se. They give instructions. Well, some do and some don't. Give specific instructions for the creation of this amino acid chain. The first one is what's called a stop codon. A stop codon means that there are no more amino acids that join this strand. And those are UGA, UAA, and UAG. I had kind of a way to remember these. You are good, you are awesome, you go away. That's kind of a way that I remember them. The other codon that has a special instruction is what's called the start codon. Every amino acid chain starts with the start codon, and it's AUG. This one should be a little bit easier. Remember what month does school start? August. So AUG is for August. Other key players in this process. The first one is ribosomal RNA, or rRNA. This is the one that reads, it clamps down and reads the mRNA and matches up the correct amino acids. The other one is what's called transcription RNA. It brings the amino acids or transfers the amino acids over to the mRNA strand. It has a nucleotide sequence on the bottom called the anticodon, and the anticodon matches up complementary to the codon in mRNA. So if we look at this, the codon is A, G, C, or U. The anticodon that would match up to it would be A would go to U, G to C, C to G, U to A. These are RNAs, so there is no T. So this is kind of a very generic example of what a tRNA looks like. You actually have the RNA strand that would be in there. You have the amino acid that's attached with it. And the bottom here are the nitrogen bases, or what's called the anticodon. So you can see that the tRNA, the anticodon, matches up to the codon on the mRNA. Translation continuing here, this process of converting the sequence of nitrogen bases on mRNA continues and starts making a sequence of amino acids. Now, mRNA leaves the nucleus and moves into the cytoplasm. The rRNA, or the ribosome, clamps down on the mRNA at the start codon, and it starts to read it. As it's reading it, the tRNAs will match up anticodons to codons, bringing amino acids to the mRNA, making an amino acid chain. This process continues until a stop codon is reached. Once the amino acid chain is released, it will fold into its functional shape and become a protein. The process of creating amino acid chains continues over and over and over using the same mRNA. You can in fact have multiple ribosomes or rRNAs reading the mRNA at the same time, making multiple protein chains. Okay, this should be the end of this video. You're going to click onto the next video to see the actual process of translation.